So, welcome back in this new session. So we will talk now with Isabelle about guidelines and examples of good practices in data management. So, as Adrian said, we are in the module 2 of this training in organic breeding about phenomics, approaches and tools for genetic resources and breeding material characterization. And you are attending to the unit 3. So as I said, guidelines and example of good practices in data management. The training outline, this outline this afternoon. So I will introduce the subject and I will talk a bit about the reproducibility crisis in science. I will give some information about the data management plan. I will explain a bit what is a DMP and the legal constraint regarding the data. I will explain also what are fair data, and I will detail the signification of this acronym. And then with Isabelle, we will give you some guidelines to manage data in a fair way. So we will talk, and especially Isabelle will talk about TD data. I will talk about standards and metadata, controlled vocabularies, licenses, and data warehouse. And to finish, we will still have a short quiz of 10 minutes. So why manage data properly? So the digital transformation is changing the landscape, involving a data deluge and a paradigm shift that need to be managed. In the past, in science, we designed an experiment, and then we collected data. And after this collect of data, there was a step of data analysis. But now the paradigm has changed. We mainly produce data and a lot of data. Then we try to organize this data. And after organization, we analyze the data and then we can share information. And this crisis of reproducibility starts from a statement. And this statement is that science isn't reproducible. A study where you can find the link to this study here with the DOI. This study showed that 19% of scientists that have been surveyed thinks there is really a reproducibility crisis. And in this same survey, a significant part of scientists admit that there have many difficulties they failed to reproduce an experiment of another scientist, but a large part of them even admit that they didn't succeed to reproduce their own experiments. Sorry. So they failed to reproduce their own results, which show that there is really a crisis of reproducibility. And this crisis is really multifactorial. And a part of the reason advanced by scientists showed that they can be related to data management. So there is a lot of reason. It is a multi-factor crisis, but a part of the explanation is related to data management. And also to the analysis and the tools, especially the source code used to do this analysis. So here I have an example with source code. So the study has been led by Kohlberg and al. and you can find the DOI on the slide. In this study, they tried to build more than 500 software from the sources they found in an article. And the study, so I won't detail the results, but the study showed that only 54% of the source code can be built. So in that case, the repeatability is okay. But in this 55%, we can see that on the right here, I show with my mouse, a part of them have been built with the help of the author of the article. A large part of this source code also needed more than 30 minutes to be built, and only 130 source code have been built in less than 30 minutes. So there is a repeatability that is okay, only for 54% of this source code, and a significant part of them needed efforts for building. In most cases, programs can be built because author refused to share the code or didn't answer to the email. This is the result that we can show here. It represents 176 source code. So the European Union react to this kind of article and to this crisis because in 2018 or so, a report of the European Union showed that producing non-fair data have a cost of 10 billion euros per year. So it's a huge amount of money. And there is now a set of law. So I won't enter in the details of this law, but this is the Open Data Directive in 2019, sorry. The free movement of non-personal data in European Union, and one important also, the GDPR for personal data. So you can find the link here to the study that showed the cost of non-fair data in the European Union. So I will be fast on that, but I wanted to make a short focus on the GDPR. So the GDPR in 5 points. So sorry, I didn't translate it here. RGPD is in French. The GDPR asks you to declare all personal data processing to the institutional DPO, the data protection officer, and assurance of compliance. You need to inform the person and to have their consent. You need to secure the media and data transfer. So you need really to secure this personal data. And you need to give to the subject a way to apply their individual rights. So they need you need to provide them a way to modify, access or delete their personal data if they want. And for sensitive data, really sensitive data, you need to implement the PAA, so the privacy impact assessment. So you need to evaluate the impact of a data loss, for example, data loss of personal data, when you respect the GDPR. So just to focus on the data protection officer, his role is to ensure the compliance of personal data processing. So usually, for instance, at ERA, we have a data protection officer, but you have to know that this person is not responsible. He's just here to ensure the compliance of personal data processing. So a word about the data management plan. So the DMP, it's a document that describes how the data of research projects or an entity will be managed through its life cycle. And the data life cycle can be divided in 7 steps. So you have the plan himself that you write at the beginning of the project. Then you will collect data or produce data. So produce data, that means you will produce data inside your project, but collect data, you can reuse some existing data. Then you will process the data. That means check, clean, validate the data, store them, and exchange the data in a secure way. Then you will analyze the data, produce scientific results, publish articles. You will preserve the data. So that means migrate data to a standard format and suitable media. Complete the metadata. So I will explain in the following slides what are the metadata. And you need to write to document this data. You will have to save them, store, submit data in a data warehouse, and have a politic of long-term data archiving. In the sixth step, you will share the data. That means you will have to define data access rights, choose a license, and then your data will be ready for reuse. So you will carry out new research, re-examine the results, the data. You will be able to use the dataset for teaching, for learning, in the respect of the license. So the data management plan, what is life cycle in a project? So at the beginning, we're setting up a project. So you will have a new collaboration. You will contact your partners, etc. And when you write your project, you will have a section on data management, and you will have to evaluate the cost of open science. So the cost to have a data manager, the cost of storage, etc. Then you will start your project, and at this step, you will setting up the data management plan. You will start the first version of the document. During the project, you will have to manage your data following the FAIR principle, and you will have to update the data management plan. The data management plan is a live document, so it can change during the project. And at the end of the project, you will be able to share your data, and respecting this rule. This is not a rule, but it is a principle, as open as possible and as close as necessary. As open as possible, because when data can be shared, we have to do it. But for example, the personal data can't be shared. So in that case, you need to make them closed. And to share your data, you will submit this data in data warehouse in open access. The objectives of a DMP is to implement best practices respecting FAIR principle. So FAIR principle means findable, accessible, interoperable, and reusable. I will detail all these terms after. The DMP will ensure the reproducibility of experiments by describing the data and how it was obtained, enabling data to be understood and reused by other partners, avoid data loss through appropriate storage, establish roles and responsibilities of everyone, respect the law and individual by clarifying the legal and ethical framework, and clarify reuse rights and sharing modality. So you have two kinds of DMP. So there is project DMP, the one that you already know if you participate to a funded project by European Union or national instances. So it is a DMP that is written in the scope of a funded project in a specific scope and fixed term, and it is mandatory. Today, all funding organization has to write a DMP. And on another end, you have entity DMP. So it will be a DMP written in in a research laboratory, for a platform, for a broader company. Any structure can have a DMP. It will be written for a larger scope and no fixed term, and it is not mandatory. But the idea, sorry, there was a question? Oh, sorry, I think it is a microphone that was not on. So the idea of the entity DMP versus the project DMP is that the project DMP will be based on entity DMP, and the entity DMP will help to prepare the project DMP. So the idea is to say, okay, I know I manage this type of data in my structure by this way. And when I participate to a project, I just have to share the section concerning this type of data to my project DMP, and you can provide information that way in your project DMP. And just some resources to write a DMP. So there is a lot of software. This is the two software that I already used. DMP Opidor, which is a French project that can be used at this URL. And it includes the main templates from funders such as Horizon Europe. It is really easy to use with a web interface, and you have forms to fill that correspond to the template of the funder that you selected. And another one is Data Stewardship Wizard, which is supported by Elixir from the European Union. And I never used this one, but it is widely used in the community of data management. So now, just I would like to share you a video. So I will try to share the sound also, because it will be better. And the idea is that you listen to this video, and that you share your idea on what is going wrong in that case with data management. So I stop sharing the presentation, and I will share the video. I try to share my Chromium. Okay. Do you see my screen?
Yes, it's a video on YouTube. Just see if your sound is sharing also.
Yeah, I start the video.
We don't hear any sound.
You don't hear any sound?
No.
Okay. I try another way.
So when you click share, you have a button include sound.
Yes, but when I try to share the Chromium tab, okay, I have it.
Hello. My name is Dr.
It's okay now.
I'm an oncologist
Works?
at NYU School of Medicine.
Hello, Dr. Judy Benign.
I read your article on B-cell function. I think that I could use the data for my work on pancreatic cancer.
I am not an oncologist.
I know, but I think I could use the data for my work on pancreatic cancer. Do you have the data?
Everything you need to know is in the article.
No. What I need is the data. Will you share your data?
I am not sure that will be possible.
But your work is in PubMed Central and was funded by NIH.
That is true.
And it was published in Science, which requires that you share your data.
I did publish in Science.
Then I am requesting your data. Can I have a copy of your data?
I am not sure where my data is.
But surely you saved your data.
I did. I saved it on a USB drive.
Where is the USB drive?
It is in a box. It is in a box at home. I just moved.
But can I use your data?
There are many boxes. So many boxes. I forgot to label the boxes.
Hello again. Thank you for sending me a copy of your data on a USB drive. I received the envelope yesterday.
You are welcome, but I will need that back when you are finished. That is my only copy.
I did have a question.
What is your question? You might find the answer in my article.
No. I received the data, but when I opened it up, it was in hexadecimal.
Yes, that is right.
I cannot read hexadecimal.
You asked for my data and I gave it to you. I have done what you asked.
But is there a way to read the hexadecimal?
You will need the program that created the hexadecimal file.
Yes, I will. What is the name of the program?
Cytosynth.
I do not know this program.
It was a very good program. The company that made the program went bankrupt in 2007.
Do you have a copy of the program?
I do not use this program anymore because the company that made it went bankrupt. Maybe you can buy a copy on eBay.
I have good news.
You again.
I talked to my colleague. She knew a person with a copy of the software.
Then why do you need me? Everything you need to know about the data is in the article.
I opened the data and I could not understand it.
If you have the program, you will find it is clear.
Well, I noticed that you called your data fields SAM. Is that an abbreviation?
Yes, it is an abbreviation of my co-author's name. His name is Samuel Lee. We call him Sam.
I see. And what is the content of the field called SAM1?
Ah yes, SAM1 is the level of CXCR4 expression.
And what is the content of the field called SAM2?
That is logical if you think about it.
What is the content of the field called SAM2?
I don't remember.
What about SAM3? Is there a guide to the data anywhere?
Yes, of course. It is the article that is published in Science.
The article does not tell me what the field names mean. Is there any record of what these field names mean?
Yes. My co-author knows what the content of SAM2 is, and SAM3. And SAM4.
Can I talk to your co-author?
I'm not sure.
I would very much like to talk to your co-author.
Well, he was a graduate student. He went back to China 2 years ago.
Can I have his contact information?
He is in China. His name is Sam Lee.
I think I cannot use your data.
You could check the article to see if what you need is there.
Please stop talking now.
Okay. So what I suggest now is during the next 5 minutes, you can share your reaction in the chat, for example, and to say what you see is going wrong in this video, in the way they manage data. So of course, everything is going wrong in this video, But specifically, what do you see that is not good in this data management? So do not hesitate to react in the chat. I will share again my screen. So do you hear me?
Yes.
Yes,
Okay.
yes.
So there is no guideline for the data. That's it. I agree with you, Estelle. Do you see something is going wrong with the software you use that to read the data? There is one copy of the data. That's true. The data were badly organized by the scientists. That indeed. The guy sends his unique copy of the data, so that's true. He doesn't have any safe data, and he doesn't have any backup of his data. The data are not public because the researchers needed to ask for the data. So I tried to read. There is much reaction now. Data format is not universal. That's it. It is not a standard because you need to have a program to read the hexadecimal format. It's not clear what can be shared or not. Indeed, contacts, there is no the research, the co-author is not referenced. We can imagine that it could have a unique identifier to identify this co-author. The scientist is reluctant to share information. We can say that, I agree. In metadata is missing. That's a good reaction. Metadata is missing indeed. And we will see further the importance of metadata. No info on the data structure, that's it. Data program wasn't available, that's true. The program is not available, and moreover, it seems we can suppose it is not a free software. So you need to buy a copy to read this data. So the data are not in an open source, in an open format. Okay, so I see that you have many ideas on that. The variable name makes no sense. You are true, Teresa. And it takes huge amount to solve the problem. 7 months. We saw that 7 months later, she had a copy of the data. Okay, so we can continue the presentation. I see that you are now aware of what is going wrong in this video. And I will talk a bit about what are fair data. So the FAIR principles are a set of guidelines that aim to make data findable, accessible, interoperable, and reusable. It provides guidelines for scientists, scientific data management, but not only scientific data management, but data management in general, and are relevant to all stakeholders of the digital ecosystem. They are aimed directly at data producers and publishers to promote maximum use of scientific data. And they focus on the ability of machines to manage data automatically with the minimum human intervention. So you can find here 2 references that explain the FAIR principles. So the second link is in French, so I'm sorry, but the first one can detail this in English. And now, if we enter in the details, what means findable? Findable means to facilitate data and metadata discovery for both human and machines. That means that data will be identified by a PID. So a PID means a permanent identifier. data are described by metadata, and this metadata includes the PID of the data they describe. And the data, to be findable, need to be submitted in a data warehouse. Accessible means to enable data access and download, which may include an authentication and authorization process. Accessible doesn't mean that data will be accessible for everybody. That means also that you provide an access to this data, but authentication and authorization are not incompatible with accessibility. So the data can be accessed via standard communication protocol. So here you have assertions that are really technical sometimes. The protocol of communication must be free and open. This protocol makes possible an access by authentication if required. So that's what we said just before. And metadata remains accessible even if data are not. Interoperability, that means to enable data exploitation and integration, whatever the IT environment you use. Meaning different operating system or different programming language. See if you access to the data programmatically. The data are described using controlled vocabularies. This vocabulary used is compliant with FAIR principle, and metadata are contextualized with links to other data. And last, to be reusable, that means to enable data to be reused for future research. That means that the metadata contains all information that may be useful. The richest are the metadata and the best it is. The data need a license for reuse, and the description of the data indicates the origin of the data. And last, the data sharing follows scientific community standards. So here you have a schema that shows what we call the 5 stars FAIR data. So producing FAIR data and open data requires different actors coming from different disciplines. So you have biologists, IT engineers, and also ontology and semantic specialists. And publishing FAIR data, so the first star starts to have just data published on the web with an open license. So it can be, for example, a PDF file with a Creative Commons license. The second star corresponds to have structured data in a structured format. Next, to have data in non-proprietary format. So to have CSV files, for example, or XML. And then you will be to the fourth star if you have a URI that identify your data. So we are talking about this notion of PID. And last, you will have your 5 stars if you have data linked to other data. Here you will work with the ontology and semantics specialist. So just here, a slide that to show the difference between accessible and open. On the left, you produce closed data, and on the right, you have open data. And between these two sets of data, you can produce FAIR data. What I mean is that if you have closed data that are not FAIR, you can't really reuse it. You can reuse data that have restricted access if they are produced in the FAIR way. Of course, you can reuse data that are open and produced in a FAIR way, but also we will consider that open data that are not produced in a FAIR way can be reused. In the example of the video, indeed, we saw that the researcher doesn't want to share his data, but even if he had published his data with the article and the data were available for download, the other researchers could not do anything with this data. It was really not well described. So now we start to give you some guidelines for data management. And I will give the floor to Isabelle, who will talk to you about TD data. So if you are here, Isabelle, maybe you can try to take control of the presentation if you can.
Yes, but I don't see the icon anymore. Maybe it's because I disconnected and reconnected. I can't see it.
I will see if I can give you the control. Okay. So anyway, I can pass your slide if you want.
Yes, please.
Okay.
Next. Yeah. So I will give you a few elements about how to make data to facilitate reuse of data, and for that we will say that we need to make tidy data. So I will first explain shortly what is tidy data and what we need that for, and then how to move from messy to tidy data. Tidy data should be complemented with tidy tools to manage and analyze the data. And everything I will present, or most of the thing I will present, is in a paper of WCAM published in 2014. And he also provided the codes with air package and the example. So it's very nice to look at this paper. And then I will shortly talk about the step further, which is to design a global data set. And additional available material for preparing and managing the data will be available at the end of Yannick's presentation. Next, please. So why do we need to tidy the data? I guess Yannick already gave good argument for that, but in 2003, Dasu and Joson found out that 80% of the data analysis was spent on the process of cleaning and preparing the data. I guess now maybe it should be much less because we are doing a more sophisticated analysis, so we spend more time in the analysis. But anyways, again, also the data we are interested in, they mostly come from the fields or from experiments. They are real-world data set, and often we prepare a framework or we prepare tables to make it easy to record the data and not to make it easy to analyze the data afterwards. And so at the end, the constraint on their organization, they are not based on to make it tidy, but to make it easy in the field. And so we get data set constructed in bizarre ways. And also data preparation is not only a first step. Usually we must repeat it many times over the course of analysis because we got additional data to include. There is a problem arising. We need to rerun the preparation, and so on. And so we need to structure data sets to facilitate analysis. Next. The principle of tidy data is to provide a standard way to organize the data values within a data set. It will make initial data cleaning easier and then facilitate initial exploration, such as finding outliers or aberrant data, then analyze the data, and also simplify the development of data analysis tools that work well together, that can pass data set from one tool to the other. And so tidy data sets provide a standardized way to link the structure of a data set, which is how it is structured, how it is organized. So it is its physical layout with its semantics, which is its meaning, what we have behind these values, these numbers. Next. So what about data structure? It's the way the data is organized in tables. So mainly how values are distributed in columns and rows. And as you can see, table 1 and table 2 are the same data, but with different layouts. Next. And a few words about data semantics. So the data set is the collection of values. It can be numbers or strings. And every value belongs both to a variable and to an observation. A variable contains all values that measure the same underlying attribute across units. The units can be plants, varieties, populations, or plots. And an observation contains all values measured on the same unit across attributes. And again, this observation can be a particular plot or a variety if you consider means or a plant or whatever. Next. So if we come back to our table 1, and so we see that it is a bit mixed up because we have for 3 varieties both we have 2 columns. One seems to be yield under organic and the other yield non-organic. And so we see that we have the values, they represent yield, maybe of a plot or average yield. And it's the same information, the same values in the 2 columns. The two headers are not really do not indicate variables, but variable name, but additional information. Table 3, which is a bit different, would be okay. Now we'll see how we can reorganize table 1. Yes, next, please. Thank you, Yannick. So what we want to do to get tidy data is to make that each variable forms a column, each observation forms a row, and each type of observational unit forms a table. And any other arrangements of the data would be messy data and will need to be tidied. So Yannik, I don't know how it's displayed, but I don't see the right column of the tables. Do you see it?
Oh.
Or is it?
I see it. So, but maybe the sharing is not optimal. You don't see
Okay.
the column yield.
Yes,
That's it.
I don't see it.
Ah, okay.
No, but it's okay.
I don't know what I can do to to have a better sharing, because I am sharing my full screen.
Okay, I will do with it. So to get a tight.
Is it better?
Yes, it's better for me. Thank you. Yes, so to get the table 1 tidy, we need to have that each row represents an observation, and so it must be the results of one farming practice on one variety. And so for that, we need to put the 2 columns to move them into rows, and then we'll have a single variable which would be yield at the plot or average yield, whatever, of a variety, and we'll have an additional variable column which will be farming practice. Okay, next. Oops.
Sorry.
Can we have the back, the slide back, please? Yes. There are 5 most common problems with messy data sets. And I won't go through the 5 of them, but only the 2 first. It may seem quite basic, but if we had a good basis, then it's easier to go further. So I will first start with column headers that are values, not variable names. Next, please. It's a bit the same kind of example as previously. We have this table where we have 3 population of plants, and we see that the values in the cells, they represent the frequency or the numbers of flowers of a particular color, and then we have the flower color as columns. So to tidy this table 5, we need to again to melt or stack the columns into rows, as we can see in table 6 on the right. And so then we need to add an additional column which will inform about the color of flower. And then we have the main column, informative column, with the frequency of plants with a particular flower color. Okay, next, Yannik, please. And then if we go to the second problem of messy data, we have in this example 2 problems, the same as the first one, when column headers are values, not variable names. And also we have multiple variables stored in one column. In this example, we have for different plant numbers that represent the diseased surface of a leaf side, it can be either the top or the bottom side of the leaf, and the leaf was recorded at 3 stages, leaf number 1, 2, and 3. So we see that we have both headers that are not appropriate, and also that in the headers we have a single variable that represents in reality 2 variables. Next, please, Yannick. So the first transformation is to melt it as previously, to put all columns into a single column with different rows. But we see that we still have a column where we have this mixed information where we have both the leaf number and the side of the leaf which has been recorded. So next, Yannick. We need then to split information into 2 columns indicating 2 variables that will allow to analyze the data. For instance, with an analysis of variance, you may test whether there is a leaf side effect or a leaf number effect in the diseased surface. Next. So, as I said, I will not go through the others problems with messy data sets. You can find all information in WCAM. Also, all the examples and the codes are available as additional material. Yes, just to say, it's important that so for the multiple types of observational units stored in the same table, the idea is that sometimes we record a lot of data in a same table. For instance, if we have a trial describing variety behavior, variety response under different practices or whatever, then we would also store like climate or meteorological information or pedological information in the same table, which will not be very informative because all rows will have the same value, and so it's best to make a separate table with meteorological or pedological information with common identifiers to be able to connect them. Next, Yannick, please. And finally, once you have a nice table with variables and rows that are tidy, then you can also sort the columns in order to make it as readable as possible. To start first with the fixed variables that describe the experimental design, such as varieties, blocks, farming practices, or whatever. So information that are known in advance, and then followed by the measured variables that you can also clustered according to their chronological recording or according to relatedness of the threads. And next. So finally, once you have tidy data sets, it's good to use tidy tools that would take these standardized formats as inputs, and that will also provide standardized formats as output. It will allow to use different tools easily and to share data sets among people doing different analysis. And again, all what you need to know about tidy data and tidy tools, you can find a lot in this article. Next, Yannick. And so final comment about making or designing a global data set. So once you have put your data set in a nice tidy way for instance, a single trial, then often you would need to analyze data coming from several trials, either that you have organized yourself, or sometimes you also want to analyze different experiments from different partners or different teams that focused on the same question and that share some varieties in common or some treatments. And so for that, it's really critical to have these standardized files, these tidy data sets. But then it's important to add what Yannick previously mentioned as metadata in order to be able to inform about all particular conditions and information that will be found in the different data sets. And so, yes, I'm finished. Then there are also good practices to share the data among partners, among collaborators, and finally, a good practice to support analysis. And so if you want to see a very a good analysis or design of a global data set and its analysis in relation to what we are doing in LiveSeeding, you can look at this paper which is applied on intercropping experiments. Next, I think I'm finished. Yes.
Thank you, Isabelle. So I can take the floor now. So Isabelle showed you how to clean and to have nice and fair data files. And now I will give you some short guidelines on the controlled vocabularies, the standard and the metadata, and I don't know by what I will finish, the license and also the data warehouse. So about the controlled vocabularies, so what is it? A controlled vocabulary makes possible to describe concepts used by a community in the same way. So the idea is to be sure that when I use terms to describe a concept, my colleague, my partners uses the same terms and to be sure that we are talking about the same things. So there is different types of controlled vocabularies. It can be thesaurus, it can be glossary, it can be also ontology, etc. And a typical example of a non-controlled vocabulary that we already made in the lab in Rai. It is the same trait written in different ways. So, for example, plant 8 with an underscore, plant 9 with an underscore, plant 10 with an underscore, plant 11 with an underscore, plant 12 with an underscore, plant 13 with an underscore, plant 14 with an underscore, plant 15 with an underscore, plant 16 with an underscore, plant 17 with an underscore, plant 18 with an underscore, plant 19 with an underscore, plant 20 with an underscore, plant 21 with an underscore, plant 22 with an underscore, plant 23 with an underscore, plant 24 with an underscore, plant 25 with an underscore, plant 26 with an underscore, plant 27 with an underscore, plant 28 with an underscore, plant 29 with an underscore, plant 30 with an underscore, plant 31 with an underscore, plant 32 with an underscore, plant 33 with an underscore, plant 34 with an underscore, plant 35 with an underscore, plant 36 with an underscore, plant 37 with an underscore, plant 38 with an underscore, plant 39 with an underscore, plant 40 with an underscore, plant 41 with an underscore, plant 8 without underscore, or plant 8 with a S to plants. So it can seem really obvious, but when you work on a project on the long term, or if you work with different colleagues, this is really the things that can happen. Especially, for example, if you have turnover in your structure, you will have newcomers that will maybe define different traits. So it really can happen. When we see this example, it really obvious that there is a need to control vocabularies, but it is not always the case. So here I just want to show you two examples of controlled vocabularies that you can use. So you have the agrovoc vocabulary of the FAO. You can find the link in the slide. And the idea here is so a thesaurus will define a list of terms that will have some hierarchical links. So for example, here you have the term agriculture, and in agriculture you have farming systems, and in farming systems you have the term alternative agriculture, and in this term you have subterms such as organic agriculture. Organic agriculture have some synonyms like organic farming. So this is the basic idea of a thesaurus, and you can find this kind of thesaurus at the FAO. So when you want to describe a farming system, so it is good to call the organic agriculture using this term. So now, if we talk about ontologies, so I'm sorry, I did not put the link to the crop ontology, but it is at the end of the presentation. So here it is an example of the wheat ontology that is hosted on the crop ontology site. An ontology is a bit different because you have indeed hierarchical relationship between concepts, but you will describe different kind of elements. For example, here you have the wheat ontology, and you will describe agronomic traits. So one of these agronomic traits, for example, is the biomass production rate. And the biomass production rate will be linked to a method that describe the way you compute the biomass. So it is a method. You describe how you take your measure. And this will be linked also to a scale. And the scale is derived by meter, square meter by day. Each term is assigned by a variable identifier. So you can see it here, CO321, 2 dots, and a list of a number. Sorry. And the ontology will be a bit different than the thesaurus because you will have links that are not only hierarchical, but objects and concepts can be used in other variables. For example, here we can see that the scale is used in different variables, and the variable will be the combination between a trait, a method, and a scale. So in this example, you have a variable name called BMPR calc GM today, and this variable is the combination between the traits biomass production rate, BMPR computation, the method, and the scale. So now, if we go on the standards and metadata, so as Isabelle said, the TD data principle will lead you to produce nice data tables. But this kind of data tables will need a set of metadata to be well described. So what are metadata? It's a report on the data. It is basically data that will describe your data, and you will, for example, store and assign to this metadata who created the data, what is the content of the data, when, where the data created, where it is geographically located, and where it has been produced, how were the data developed, and why were the data developed. So this is the basic description of metadata. So at this step, we can say that we have rich metadata. And why we will use a standard. So using a standard will allow to analyze, compare, and exchange data. You will be able to publish datasets in international data warehouse. So really the standard will make it possible to share the data and to make them reusable. And the metadata standard will describe data richly and accurately using the same vocabulary as the rest of the scientific community. And it will make your data interoperable and enable other systems to use them. So in a way, describing basic metadata is the minimum, but using a metadata standard that will be really specific of your domain will allow you to describe your data set with rich metadata, with a lot of attributes. So here it is just an example of text formats that are frequently used for metadata. So the CSV format, it is basically a text format with fields that are separated with a comma. The advantage of this kind of file, it is human readable, while the XML and JSON format that you can see here in the center and at the right are really good format if you want something that is machine readable, but edit this kind of format for human is not something easy. So sometimes you can have also some tools that will make it possible to edit this kind of format, and you save it as a XML file or JSON file, but you will have an interface that will make it possible to edit this kind of file. Just a word about the XLS format. This is not a standard format because XLS was the Microsoft format that were not an open one, but XLSX is a format in XML, so it's okay. So just be careful with that. XLS is not really a standard format, but XLSX can be used. And now about the metadata standard, I just would like to make a focus on the MyAPI standard format. MyAPI means minimum information for plant phenotyping experiment. It is a standard of metadata for a phenotyping experiment. So I won't enter in the detail of the MyAPI standard because it will need a whole training. So it is here just the basic information and just to provide you an interest for this metadata standard. So the standard is quite simple because it is the template is an XLSX file, so it is human readable. You can really edit it and understand it. When you download the template from the website of the MyAPI if you download the template file from the MyAPI website, you will see that you can download the file, and it it is a spreadsheet file with different cheats. And you can describe the investigation. So the investigation will correspond to the project you are working on. You will describe the study. The study will be a set of trials that you have in your project. You will describe the person that will lead the study. You have a sheet with the data file. So here I will give some information on that. You describe the biological material. So basically the genetic resources you are using. And you will be able to describe environment variables, so information about your environment, cultural practices, the experimental factors. If there is an event during the trial that occurs, you can fill this kind of information, etc. And I will give here details on the three main fields that are interesting for us. The observation unit. So the observation unit will describe the biological material that you use, but at a particular location. So you will say, I will test this variety on this plot, and you will fill the coordinates of the plot. The observation variable. Here you will define the variables of your study, what you will measure. And as I show for the crop ontology, the observation variable will be a combination between a trait, such as plant 8, a method, so how you measure the variable, and a scale. And so as soon as you have described all these elements in observation unit and observation variable, you will be able to produce some TD data files like Isabelle described just before. And your TD data file will be a matrix between your observation unit and your observation variables. And that way, if you have clean tidy data files and a MyAPI file with your metadata that are richly described, then you can say that you have a fair data set. And the advantage of the MyAPI standard is that it includes some established standard like the multi-crop passport description, which is a set of fields that describe genetic resources, and the crop ontology that I talked about just before. So now a short focus on the licenses. So why to choose a license? So the license gives a framework for data sharing and reuse. It allows users to be granted a certain usage right. It may include restriction on use. So the contract of the license will say what you can do with the data. It is strongly recommended in all cases to clearly display the related rights. Of course, you should not diffuse some data without a license, because without a license, your data, you lost the ownership of your data. So everybody can do what you want with your data, and even don't need to cite your work. So what are the recommended license? Mostly the widely used license, because it is a matter of confidence. The license that are compliant with other existing license to ease data aggregation. And you need to take into account the potential of the data and the restrictions applied. For example, in France, we have a license created by Etalab. And sometimes we need to publish data under this license, but mostly if we publish data for an international public, we will use more Creative Commons license. So the Creative Commons license, so you just have here a table that summarizes the different clause of the Creative Commons license. So just to summarize this different clause, the CC0 license means that your data will go in the public domain, and this is not really recommended because it is as you publish your data with no license. That means that the people who use your data don't need even to cite you. The AND clause is not also recommended because it makes impossible to modify, adapt the data, and that means that someone that want to use your data even can't aggregate your data with another data set. So it is a big restriction to the usage of data, so it is not recommended. And what is not recommended also is the clause of no commercialization, because you prevent any commercial use. So sometimes you can feel that it is a good idea because you don't want that your data can be used for commercial use, but commercial use includes any non-profit action, such as a training that will need an inscription fee. So often it is not really what you want. So non-commercial use is not recommended at all. So about the Creative Commons, the two licenses that are mostly recommended are the CC BY and the CC BY SA. And I wanted also to make a short focus on the license on code source. So even if you do not have code source is considered as a kind of data. And you have two main type of open source license for source code. The permissive one, that means that this kind of license, you can modify the code and change it. You have to be cited, but the resulting code doesn't need to be shared. It can become a proprietary code. So of course, your code that is distributed under a permissive license remains open. But if another developer use your code and modify it and add some lines of code, he can publish this one under a proprietary license. The other kind of license are the copyleft license. And this license means that you can modify the code, but you must publish it under the same term of license. So in that case, we talk about viral license, and all those kind of license are good to use, but it will really depend on the context of your code. And now the data repositories or data warehouse. So you will have two kinds of data warehouse. You will have some thematic one that are so the positive aspect of this data warehouse. It is accurate for data description and metadata because you will have really rich metadata in this kind of warehouse. And so your data set will be of good quality. They will be really fair. And the problem is that it does not exist for all data types. So for example, we don't have thematic data warehouse for phenotyping data. And the publication of data in this kind of warehouse is really time consuming because you have a lot of metadata to fill. And the general data warehouse, so the positive aspect, so it is the contrary of the thematic warehouse. So you will be able to publish all data types. You would not have many metadata to publish, and you can publish really fast. But the problem is that you will have a poor description of your data. Maybe sometimes it will use non-standard format, and in general way, you will be less fair. So here you have some examples of data warehouse. I won't cite all of them, Maybe one of that is widely used today is Zenodo. Dryad also is mostly used. And in France, we have the Recherche Data Gouv repository. So I think it's possible to publish data when there is collaboration. But I don't know if there is some other national data warehouse. Probably, but I don't know. I don't have an exhaustive view of all this data warehouse. So just some tips to choose a data warehouse when you want to publish some data. So this is my opinion. You can follow this criteria to choose your data warehouse. First, try to choose a thematic warehouse, especially if it is an established one, such as you have, for example, the ENA, European Nucleotide Archive for sequence data. So it is managed by the EBI, which is a European institute. So it is something established. So when you have thematic warehouse, maybe give the priority to this warehouse. Anyway, you can choose a national or institutional warehouse if you have one in your country. And if you don't have warehouse that fit this criteria, I think take into account this criteria. Try to find a warehouse that is open source, that have a general scope, that provides a DOI for your data sets, and that is robustly funded. This last criteria is almost important because you have to take into account that even if you find a data warehouse that is based on an open source project and that provide a DOI that mean it seems to be established, etc. If it is not robustly funded, your data can disappear from a day to another. So, for example, I'm not sure that it is the case today, but Zenodo was funded on a European project. So European project, of course, that means that Europe fund Zenodo, but it is fixed term project, so it is not permanent. So I think that Zenodo now have a more robust funding, but it was not necessarily the case in the past. So be aware of this criteria to choose your warehouse. And now, so what I suggest to pass to the short test like this morning. I will copy paste the link in the chat. And I will also pass the email where you can send the answer. So I think that we can give 10 minutes for the chat, and after this for the test, sorry. And I think that if you have any question after that, we can have some few minutes for questions. So we can restart in 10 minutes at 38.
Yeah.
So just to conclude, I wanted to say to share some key message. So just try to be fair with your data. Use tidy data, standard and controlled vocabularies to increase the quality of your data. And share your data with appropriate license and warehouse to make them reusable. And if we have some few minutes, if you have any question, I think that you can share it in the chat like this morning, and we can answer to your question with Isabel. And if there is no question, I think it is the time for a break if Petra and Adrian, since it is a time.
Okay. So if everything from your side, Yannik?
Yes, just also, I didn't say it, but there is a list of resources at the end of the presentation. So, we will share the support. So, but I have nothing to add.